Hi guys! Now let's learn about meiosis. These are the learning objectives that you need to complete at the end of this lesson. At the end of the lesson, students should be able to define chromatid, homologous chromosome, synapsis, bivalent, tetrad, chiasma, crossing over, and centromere. Besides that, students also need to be able to state the stages of meiosis I and meiosis II. Lastly, students need to be able to explain the behavior of the chromosomes at each stage. Meiosis. Meiosis is a type of cell division whereby the 4 daughter cells produced will be genetically non-identical from each other and from the mother cell. The daughter cells will also have half of the number of chromosomes as compared to the mother cell, or in other words, the daughter cell now becomes haploid. Meiosis can be divided into two phases. These are meiosis I and meiosis II. Both meiosis I and meiosis II have 4 stages. These are prophase I, metaphase I, anaphase I, telophase I, followed by prophase II, metaphase II, anaphase II, and telophase II. Depending on the species, cytokinesis will separate between meiosis I and meiosis II. Since all the events taking place in meiosis II is the same as mitosis, meiosis II will not be discussed in this video. Prophase I. During prophase I, the decondensed chromosome in the form of chromatin begins to coil into individual chromosomes. These chromosomes can be seen clearly because they appear shorter and thicker, as you can see in the diagram. As the chromatin coils, the nucleolus disappears. Next, pairing up or the synapsis of homologous chromosome will take place, forming a bivalent or a tetrad. Once bivalent is formed, crossing over will take place between non-sister chromatids. The diagram is showing non-sister chromatids. The point where crossing over takes place is known as chiasmata or chiasma. Next, the nuclear membrane will disintegrate. The centrosome will begin to migrate to the opposite poles and radiate spindle fiber. That concludes all the events that take place during prophase I. Metaphase I. During metaphase I, the centrosome has arrived at the opposite poles. The bivalent now line up at the metaphase plate. Metaphase plate is an imaginary line at the center of the cell. It is not a discrete structure. The spindle fiber from one pole will attach to one of the homologue. Meanwhile, the spindle fiber from the other pole will attach to the other homologue. In anaphase I, the spindle fiber shortens or contracts. This causes the homologous chromosome to separate and begin to migrate to the opposite poles. The arrival of the separated chromosome marks the beginning of telophase I. Once the separated chromosome has arrived at the opposite poles, they will begin to uncoil and form chromatin again. Once this happens, each individual chromosome will no longer be able to be seen. When chromosomes begin to uncoil and form chromatin, the nucleolus will reappear. Lastly, the nuclear membrane will reform. So those are the events that take place in meiosis I. It is important for the student to note that at the end of meiosis I, the daughter cells produced are already haploid, or having half of the chromosome number as the mother cell. Let's take a look. This is the mother cell. This mother cell has 6 chromosomes, 1, 2, 3, 4, 5, and 6. By the end of meiosis I, each of the daughter cells only have 3 chromosomes, 1, 2, and 3. So therefore, the halving of the chromosome number already takes place at the end of meiosis I. These are the importance of meiosis. Meiosis is important in the production of gamete cells in sexually reproducing organisms. Meiosis will result in variation via crossing over and also independent assortment. This is independent assortment. When the homologous chromosomes line up at the metaphase plate, there are many possible arrangements of the homologous chromosomes. In this case, there are two possibilities because it involves two pairs of homologous chromosomes. The first possibility is the blue is in one side, the red is all in one side. In the second possibility, it's mixed. So therefore, there will be 4 types of daughter cells produced, thus creating variation. Meiosis is also important in halving the chromosome number in gametes, so that the species chromosome number remains constant in sexually reproducing organisms. Can you imagine what would happen if we produce diploid gametes? Humans have 46 chromosomes, so therefore, if 2 individuals come together and both of their gametes are diploid, the next generation will have 92 chromosomes. When 2 individuals from the second generation get together and they are also producing diploid gametes, the third generation will have 184 chromosomes. And if this continues to happen, the species chromosome number will continue to increase exponentially.